Hello, and welcome back. We're going to go over another example of a monohybrid cross, okay? This time we're going to define the parental generation or the parental flowers as being homozygous recessive, okay? So we've got one plant that has purple flowers, one plant that has white flowers. Both are homozygous. This one would be homozygous dominant because both of the letters are capital. This would be homozygous recessive because both of the letters are lowercase. Okay? So we're going to draw our Punnett Square. Woohoo! Squeakiness. You would think I would have replaced that marker, but no, we're just going to deal with the squeak. Okay? So again, our parental generation, we're going to assign one letter in front of each box, okay? So we'll put the dominant allele up top, okay? Capital F, lowercase f, okay? We fill in our Punnett Square. Again, we look at our alleles. Remember, the dominant allele always goes first. So we have capital F, lowercase f, capital F, lowercase f, capital F, lowercase f, capital F, lowercase f. Okay? So our F1 generation, in this case, is going to be very uniform. And that's because both parents were homozygous for the particular trait that we were tracking. Okay? So when we have both parents being homozygous, our offspring are going to be all heterozygous. So they will get one dominant allele from one parent, all right, the father, one dominant allele from the mom, or I'm sorry, one recessive allele from the mom. So we get a recessive and a dominant. Our F1 in this case, okay, 100% are going to be heterozygous, okay? Phenotypically, they're all going to look the same, okay? So we'll also have 100% that are going to have purple flowers, okay? So that's how we're going to interpret that particular Punnett Square. And why this is important is because how we set up these Punnett Squares, this is essentially what Mendel was tracking with the traits with the P's, okay? And what that led him to was Mendel's first law, okay? And that first law is called the Law of Segregation, okay? And what that basically, what Mendel was explaining with his law of segregation is that each gamete can only contain one trait. Okay, and he called it a trait. In this case, you know, we now know we're tracking genes, but what the point of that was is with our alleles, if you have two different alleles, any particular egg or any particular sperm can only have one of those, okay? And that's what we set up here, okay? When we set up the Punnett Squares, when we set up the letters in front of the boxes, essentially, this would represent the gamete of one sperm. This would represent the gamete that would be available for the other sperm if our capital F's are dads. If our lowercase or our recessive alleles are mom, the egg would have either the lowercase f or the lowercase f. Okay? Now, if we change our gametes, if we take the F1s, which is essentially the cross I explained in the previous video, right? If we take our F1s, we can either have the dominant trait or the recessive trait for the dad, the dominant trait or the recessive trait for mom, okay, if we cross two heterozygotes, okay? We fill in our Punnett square. This is going to look just like the Punnett square in the previous video, okay? Our F2 generation then would have these particular genotypes, okay? Again, segregating, the point I wanted to make with this is you're going to segregate into the gamete. The egg can either get the dominant allele or it will get the recessive allele, depending on which chromosome segregates into that gamete during meiosis, okay? So with that, Mendel explained the law of segregation. So in those gametes, you can only have one trait in each gamete, all right? Now, there's one big caveat on this, because I mentioned before this is going to be true for genes that are unlinked, okay? We're not going to really cover in this class how linked genes are transmitted, but if genes are linked, linked genes very often are going to travel together, because essentially when a gene is linked, squeakiness, linked genes, we'll call this gene 1, and we'll call this gene 2, are going to be physically very close on an individual chromosome. Now what ends up happening, and how you can get some odd stuff when you follow linked genes, is if you get any genetic recombination that occurs. So remember when we talked about crossing over? If you have an event of crossing over, when we were talking about meiosis, in between these two genes, then they will look like they're no longer linked, okay? So there's some weird stuff that goes along with following linked genes. I'm not going to have you follow a Punnett square with linked genes in this class. If you take an advanced genetics class, that would be something that you would learn more about. I do want you to understand what the concept of linkage is, okay? So just keep that in mind, all right? Our next video, we're going to go over what happens when we follow two traits, okay? Because right now, with the monohybrid cross, remember, monohybrid cross, we are following just one trait, okay? Next video, we're going to go over what happens if we follow two traits, all right? If you have any questions, let me know. Thank you for your time.